Guys, welcome to 10.1 and 10.2. We are talking about photosynthesis, the light reactions. So here in photosynthesis, we are talking about this amazing process that has probably only evolved once. It is absolutely mind-blowing that it even evolved once to begin with. And so we go back to Lynn Margulies's theory of the endosymbiotic theory, understanding that the first chloroplast, or the beginnings of a chloroplast, came from a photosynthetic bacteria or a cyanobacteria being engulfed by a host cell. And so there are chloroplasts in a number of organisms, for example, algae. And so here, chloroplasts in a leaf, so we're going to be talking about leaves right now. The mesophyll tissue is where most of our chloroplasts are. They are very numbered. There are very numerous. There's 500,000 chloroplasts per 1 millimeter squared, so they are very, very abundant. And right here we can see this is the outside of the leaf. So this is sort of a cross section, and this is the stomata. And this is where the gas exchange occurs. And so the CO2 goes in and the O2 comes out. And then this is the vein in a leaf. And this is what's going to be bringing in the water from the roots, which we know gets there through transpiration. And we understand that because the osmolarity is going to decrease as it goes up the tree. And the sugars are going to be moved and cycled there. And so that explains all of the conduits, or everything that we need, the way that everything that we need is going to get to the chloroplast so that photosynthesis can occur. And this is a close-up of a chloroplast. Here is our outer membrane and our inner membrane, and we can see that this is similar to a mitochondria, except where there's no cristae here, so this is kind of more smooth. And instead of the inner membrane space, we're talking about, we call it the stroma. So the inner membrane space is called the stroma. In the stroma, we find thylakoids. A stack of thylakoids are called a granum. Many granum are called a grana. And inside of this thylakoid is where the chlorophyll is. Those are the pigments in which that allows photosynthesis to occur. And so if you were to, so light needs to penetrate through 1 to 3 lipid bilayers in order to get into the place where the chlorophyll exists. Here's our photosynthesis equation. Hopefully you have committed this to memory years ago. 6 CO2 plus 6 H2O. React to create C6H12O6 plus 6 O2. The photosynthesis equation was determined in the 1800s. However, this has been simplified to make connections to cell respiration a little bit more easy. And one way that it's been simplified is that glucose is not actually produced in photosynthesis. It is produced after photosynthesis, but what is produced in photosynthesis is G3P. G3P is a 3-carbon molecule, which is also a sugar, and those are combined to make a glucose. And so more accurately, we can show that our process of photosynthesis looks like this, a CH2O instead of a C6H12O6. So initially, scientists believed that O2 that was given off by plants came from the carbon dioxide, because water is very stable. There was no way that water was being split. There's so much energy is released when the oxygen and the hydrogen combine there. It's a very stable molecule. And so everybody just assumed that it was the carbon dioxide being separated. And a scientist, Van Niel, was investigating photosynthesis in bacteria. And they make their carbohydrates from carbon dioxide. However, they don't release oxygen. And so where is that oxygen going? Or where did it come from when it was produced? So CO2 is not separated into carbon and oxygen. He determined in one group of bacteria that the hydrogen was stripped from the hydrogen sulfide instead of the water. And so perhaps the oxygen from plants is being stripped away from the hydrogen as well. So what he did is he conducted an experiment. This experiment was using a heavy oxygen isotope called O18. So it's the O18 isotope. And he put O18 into carbon dioxide. So here is the carbon dioxide with O18, the heavy water, or the heavy carbon dioxide. And he put oxygen, heavy oxygen, into the water, or the H2O. So this is one test group, and this is another test group. And when the carbon dioxide was released to a plant, the resulting oxygen did not contain heavy oxygen. And so when the water, the heavy water was given or introduced, the plant that the oxygen released contained the heavy oxygen. And so that was the conclusion, is that the water is being stripped, or the hydrogen is being stripped of the oxygen, and is released as a byproduct, which makes photosynthesis even cooler. This is why biologists love photosynthesis. It's just a cool process. And so photosynthesis is not the exact opposite process of cell respiration. We break things down sometimes so basically that they become untrue, but it helps us to understand them. It is not the exact process, but the general reactants and products are reversed. It is the redox reactions are a reverse of a combustion reaction. So I love this schematic, so we're going to use this. Even though this is methane, I want you to pretend that this is a sugar molecule, an energy molecule. And so here, during cell respiration, what happened was oxygen, the greedy little atom that it it hogged all of those electrons from everybody, from the carbons, from the hydrogens. It just took the electrons away, and it stripped the energy from the molecules. And what's happening in photosynthesis is that it's just being reversed. These electrons are now going back into products in which the electrons are shared more evenly. So they're going back to O2. It's going back to the sugars, which contain more energy, in which the electrons are shared more evenly. So the big idea, let's go over the big idea so that when you get lost in details, you can just come back to the big idea. Photosynthesis occurs in two stages. The light reactions, photo, so think of this as the light reactions. Light is required. You need light in order for this to happen. And the Calvin cycle, this is the synthesis part. This is the building of the sugar molecules, or of the G3P molecules. So photo is the obtaining the light, and then the synthesis, or the Calvin cycle, is the synthesis part. And so during the light reactions, light energy is converted into chemical energy. Water is used. It provides the hydrogen ions, or the hydrogen and the electrons. Remember that the oxygen is split and it is released as a byproduct. The light provides the energy to reduce, for the reduction of hydrogen and the electrons from water to NADP plus to make NADPH. So we are now creating NADPH. So think of this as NAD from or NADH from cell respiration, but just add a P in there, and you can think the P stands for photosynthesis. I always think of that, and it stands for photosynthesis. When in fact it's nicotinamide adenine dinucleotide phosphate, or phosphate. But because there's another phosphate group, but you can just let it represent the photosynthesis. They also generate ATP using photophosphorylation. This is really similar to the electron transport chain. It's a process using chemiosmosis. It occurs during the process of the light reactions, and it's very similar. So we'll talk about that. Calvin cycle, the synthesis. Carbons are incorporated into organic molecules. We call this carbon fixation. And then the NADPH is then used to reduce these carbon molecules, or to add the electrons and the hydrogens to these carbon molecules to make them into sugars. So we're taking the energy from the light, we're bringing it over to the Calvin cycle, and we're taking those hydrogens and electrons and adding them to sugars. And that is the big idea. We are making NADPH, we're sending it over, and we're using that NADPH to fill up or to fuel up carbon molecules. So it's really opposite. It really is opposite of cell respiration. Light reactions. So light energy travels 93 million miles from the sun in about 9 minutes, which is really cool to think about. And the wavelengths that travel from the sun occur between our infrared to UV. So that's what we get from the sun. And the waves that we get are electromagnetic waves, and we call this the electromagnetic spectrum. And the light that we can see, or the electromagnetic waves that we can see, we call visible light. Those occur in 300, those wavelengths are 380 nanometers to 750 nanometers. Red light has the longest wavelength, so this is the longest, and purple or violet has the shortest, so that is the greatest amount of energy. The least amount of energy, the greatest amount of energy. And so when light meets or enters the atmosphere, it is either going to be reflected or absorbed. And so when it reaches a chlorophyll molecule, it is going to be absorbed. And there are a couple of different chlorophylls. We have chlorophyll A and chlorophyll B, and there's actually more, but we're just going to talk about A and B and carotenoids. So why are there multiple? Why are there so many of these pigments? Well, because each of these pigments absorb light at a different wavelength. And so if we have a more diverse population of chlorophylls, then we're going to be able to absorb more of the light, or use more of the light that's coming down from the sun. And notice how much of a difference there is between chlorophyll A and chlorophyll B. It's just right here we have just this small molecule over here that is added onto it, that is going to change the way in which it can absorb light. And carotenoids offer photoprotection. So they are within the thylakoid membrane, and they can absorb energy that would otherwise hurt the chlorophyll or negatively impact it. And it also can collect energy from different wavelengths and pass that on to the chlorophylls. So they are very important. Actually, they're also very important in our diet. We need carotenoids to survive. And so when light energy is absorbed by pigments, it doesn't just disappear. Energy is not something that just disappears. It's actually a thing, right? So here are our photons incoming, and what happens is when these photons hit the electrons, it moves the electron to a higher energy state. And then in a billionth of a second, it drops back down. But the photons that are absorbed by chlorophyll A or by chlorophyll B are specifically and only those that are equal to the difference between the ground state and the excited state. So in a very, very basic way to understand this, that is so simple it might not be true, or it is not true, but it'll help to understand that if you think the size of the wavelength here is going to be the size of the path of the electron. So how far out is it going to go? What is the difference between the ground state and the excited state? That's going to indicate what the size, the wavelength of the photons or the electromagnetic waves that are coming in. And it can only absorb those that are that size. Let's look at these chlorophyll molecules that are in the thylakoid membrane. So they are contained in these photosystems. A photosystem, think of a protein complex. It is a protein complex, just like in the electron transport chain. So multiple proteins, there's cofactors, et cetera, et cetera. So we've broken down these photosystems or these protein complexes into two categories. One is the light harvesting complex. So that's here and here and here, and this is where the light is going to be absorbed by the chlorophyll. These are chlorophyll molecules, and the energy is passed on until it reaches the reaction center complex. So here we're talking about the reaction center complex, and here we have a couple of chlorophylls that are. The chlorophyll molecule is the same as the molecules out here, except there's two of them, and then the environment is different in the interaction with molecules in the reaction center complex, which allows it to respond differently to energy. But the molecules are the same. And so let's then look at, there is a notes assist on PowerSchool or our class page, and I recommend printing this out and labeling everything that you see here so that as we go into the next part of the lecture, it is going to make more sense. It'll be easier to learn. So I recommend taking a pause and doing that. The light reactions. The thylakoid membrane is populated with two types of chlorophyll. So in two different photosystems. There's photosystem 1 and there's photosystem 2, or PS1 and PS2. And so let me show you. There's a picture of this here. Here's our thylakoid membrane. And we have chlorophyll 1 and chlorophyll 2, but we have right here our photosystem 2 is what we're going to talk about first. And here's our photosystem 1. And so these are all part of the light reactions, all part of the same process. And chlorophyll is our molecules in each photosystem, but the way that they interact with the protein accounts for the slight differences in the light absorbing properties. So the chlorophylls in both of those photosystems are going to be absorbing light at different wavelengths. So the chlorophyll in, let's see, the chlorophyll A in photosystem 1 is going to be absorbing at 700, so right here, 700 nanometers. Whereas at photosystem 2, it's going to be at 680, so a little bit further down. And it's not that the molecules are different, it's just the way that they are interacting with the protein and the other parts of the protein that change the way that they can absorb light. And so here, let's start talking about the process of the light reactions. We're going to follow an electron. We're going to follow its path, similar to cell respiration. And so during the light reactions, NADPH and ATP are produced, and these are used to fuel the Kelvin cycle. We call this the linear electron flow. We're calling it the linear electron flow because later on we're going to talk about instances when it isn't so linear. So notice the linear shape of the electron flow here. So in step 1, a photon of light, so here's our photon of light, is going to strike one of the light harvesting complexes of photosystem 2. So it's counterintuitive that we start with photosystem 2, but we do because it was discovered second. And what's going to happen is that energy, so understand that it is the energy that is being passed. It is not an electron. It is energy that is being passed from one chlorophyll molecule there. So this one gets excited and it passes the energy onto this one. This one gets excited and it passes the energy onto this one. This one gets excited and it passes the energy onto this one, and so on until it reaches the electron pair of chlorophyll. and is transferred from P680 to the electron acceptor. So what happens here is that an electron, so when that energy is transferred here to the P680, or to the, also known as the chlorophyll pair, that energy is going to excite an electron. And so here, understand that these chlorophyll molecules were excited. The electrons went to an excited state, but then came back down, and it passed that energy on. Here, when it gets to P680, the chlorophyll pair, here, it's going to send an electron to an excited state, but because of the environment, because of the pair of molecules, because of what it is surrounding it, instead of that electron coming back down to a ground state, it is sent to another molecule. It is sent to an electron acceptor. So remember in cell respiration that oxygen was an electron acceptor. And so now here, understand that P680 is missing an electron and it needs to be refilled. Here we are at step 3. An enzyme is going to catalyze the splitting of water. It's going to split it up into 2 hydrogen ions and 2 electrons. So the 2 hydrogen ions are going to be released into the thylakoid space. So let me show you, give you perspective. So here is our water, and it is going to be split. Oxygen combines with another oxygen and then become O2, and then the hydrogen is released, it stays in here, which is going to be important as we talk about chemiosmosis later. So the hydrogen ions stay in here. And the 2 electrons, they refill the very electronegative P680, so, or those 2 chlorophyll molecules. An oxygen atom is immediately combines with another oxygen and is released to the environment. The electron that was supplied by the water is accepted by the P680, and then it is passed from photosystem 2 to photosystem 1 via an electron transport chain, so an ETC, just like we've studied in cell respiration. And as the electron passes through the electron transport chain, a hydrogen ion is pumped into the inner thylakoid space. So it is passed. So here the water is stripped. So here our water is stripped, oxygen is released, and the electrons are then going to be passed down to the P680. P680 is very electronegative. The second that an electron is free, it's gonna pull it in, and then the energy from the light that's been passed down is going to send that electron out into an energized state. That electron is then going to be accepted by a primary electron acceptor, and it's going to be passed from plastoquinine to a cytochrome complex, and then plastocyanin, all the way down till we reach our second photosystem, or P700. I am having you guys learn or know these words, the plastocyanin, the plastoquinine, because things are going to be different when we're not talking about the linear model. So the hydrogen ion on the inside of the thylakoid is used to make ATP. So when the electron passed through the cytochrome complex, it allowed for a hydrogen ion to pass through the membrane into the inner thylakoid space. So here, the electron passes through the cytochrome complex over to photosystem 1. And as that happens, as it is reduced, it is going to allow a hydrogen. So now we are building up hydrogen ions in the inner thylakoid space. Why is this important? This is chemiosmosis. And so these hydrogen ions are now going to pass through an ATP synthase, and they're going to produce ATP using photophosphorylation. So photophosphorylation is not a scary word. Using light to add a phosphate group to an ADP. That's it. And so we just have another ATP synthase right here. The Calvin cycle, oh, let me go back up here. So the hydrogen is used for chemiosmosis. And so meanwhile, we've sent the electron over here to P700. It has a light harvesting complex. And what's going to happen is light is going to be absorbed just like it was over in a photosystem 2. And it's going to send that energy, and then that electron is going to be, so the electron here is going to pull in that electron, and it's going to send that electron to another electron receptor. Once the P700 pair of chlorophyll A molecules sends the electron up, it is now ready to accept another electron. So the 2 electrons per 1 water molecule, but there's going to be a constant flow of electrons. So it's constantly being restocked. The electron that has been passed down is going to be passed down another electron transport chain, but there's no chemiosmosis in this one. It's going to be passed down to the ferredoxin, and then it's going to be passed down to NADP plus reductase, where it will then, oh, I'm on the wrong slide, where the NADP reductase, where it's going to transfer 2 of the electrons, so 2 electrons from the electron transport chain. So these electrons have passed from the water all the way down this process. It's going to take 2 electrons and a hydrogen ion from the stroma and add it to NAD plus to create NADPH. So that name, NADP plus reductase, makes sense because we are reducing NADP plus. The electrons in the NADPH are at a higher energy state in the NADPH molecule than they were in the original H2O molecule. So we have moved those electrons closer to they're being shared more evenly than they were at the beginning of the process. And so that is the end of this lecture. I hope I can answer any questions that you might have.